Let's continue here with indirect agglutination tests. And in panel A, you have an example where you have latex beads that are coated with antigens in sort of this golden color right here. And the agglutination, if it's present, it indicates the presence of antibodies such as the IgMs that are shown here. So you have IgMs right here, and if you see an agglutination reaction, you know that the antibody has found its target, which is these coated latex beads. In panel B, you have the latex beads, and now they're coated with antibody, as you can see here in green, these Y-shaped structures. And now the agglutination reaction indicates the presence of the antigen. So if there's an agglutination reaction, it means this antigen that's binding to this antibody is present, and you get this clumping reaction. In a viral hemagglutination test, you're going to have red blood cells that will interact with viruses to produce the clumping reaction. The hemagglutination in this case looks just like a blood typing test, but in this case, the red blood cells will be binding to viruses. Moving on to neutralization reactions. In this case, you may have an antigen-antibody reaction where the harmful effects of an exotoxin or a virus are blocked by antibodies to the toxin, and I'll show you that in the next slide. Viral hemagglutination inhibition test is used for subtyping of viruses. For example, viruses in RBCs or red blood cells are mixed with the patient's serum. If the serum contains antibodies to a virus, they neutralize the virus and inhibit the hemagglutination. But here would be sort of this neutralization test. So here you have toxin molecules in a cell, and the cell, if it gets damaged by the toxin, that's what it looks like. The cell gets killed. Now here are the toxin molecules, and you have antibodies, the antitoxin. So antibodies to the toxin, which is the antitoxin plus the cell. And now the antibodies they neutralize the toxin, and the cell is no longer damaged. So the cell stays intact. So that would be a neutralization reaction because the antibody neutralizes the toxin. And down here, the effects of a toxin on a susceptible cell, neutralization of the toxin by antitoxin. Here you have some red blood cells, and then the antiviral antibodies from the serum plus the virus. And now the viruses are neutralized and the hemagglutination is inhibited because the antibodies will bind the viruses and neutralize the virus, and nothing's going to happen. Another application is the complement fixation reaction. So when you're talking about complement fixation, you're talking about that there has been an antibody-antigen interaction, and that interaction has changed the stem region of an IgG, and now the first component of complement can bind to it. So that means the complement serum protein binds to and is fixed to the antigen-antibody complex. It detects a relatively small amount of antibody, and it works for antibodies that do not work with precipitation or agglutination reactions. So here is a complement fixation test. And just here, over here in the left side, you have a positive test. That means the antigen and complement interact with serum antibody against the antigen, and then basically you get no hemolysis in this reaction. Over here with a negative test on panel B, we have the antigen and complement. There's no the serum does not contain any antibody, and so now the antigen antibody interaction is not there, and complement does not get activated. So now you get hemolysis because the complement, they're not coated, these red blood cells. Moving on to fluorescent antibody techniques. So fluorescent antibody, a lot of times it's abbreviated as FA, fluorescent antibody. Basically, you're combining an antibody with a fluorescent dye for detection purposes. You can do this with a direct fluorescent antibody test or indirect fluorescent antibody test. In the direct test, you identify a microorganism in a clinical specimen. In the indirect FA test, you detect a specific antibody in the serum. And so here's an example: the anti-human immune serum globulin, anti-HISG, is added and will react with any antibody in the serum if the result is positive. Here's a diagram showing you results of fluorescent antibody testing. First, let's take a look at the direct fluorescent antibody test. So here you have group A streptococci from somebody's throat swab. And you have fluorescent dye-labeled antibodies. So these guys are glowing fluorescent. And once they find their target right here, then you have fluorescent streptococci that you can detect in a microscope. In the indirect fluorescent antibody test, now you are taking here Treponema pallidum from laboratory stock. Here, the example, you have specific antibodies in serum of a patient, and now you're taking this antigen that you have from a lab stock plus the antibody that is present in a patient that has been infected, and these antibodies will bind to Treponema pallidum, and now you have fluorescent dye-labeled anti-human immune serum globulin, which will react with any immunoglobulin, and now you have a fluorescent spirochete test here, and that would be a positive test for the presence of Treponema pallidum. Very popular these days are fluorescence activated cell sorters. That technique is abbreviated FACS, and it basically allows you to separate out cells in a sample. So a laser beam strikes a droplet containing cells, various cells, and then the detector determines size and fluorescence of the surface of the molecules. And in the end, you're basically separating out cells. And this is used a lot these days. This is very popular. Let's say you want to have a scattergram of different white blood cells from a patient, and you want to separate out all of these different white blood cells. You do a fluorescent label for different white blood cells, and then you separate them out with a FACS. Here you can see an example of how the FACS cell sorter works. You have fluorescently labeled cells, those are the orange ones. Then you basically run them single file in front of a laser beam, and then the laser beam will sort out the fluorescently labeled cells from the non-labeled cells, and you're sorting out the fluorescently labeled cells into one tube and the non-labeled cells into the other tube, and that's how you're separating out cells. Moving on to a very important technique that's called ELISA. So that's the abbreviation for enzyme-linked immunosorbent assay. There are two versions of that. You can either do a direct ELISA or an indirect ELISA, and you do have a labs to lab that is assigned for this week, so be sure to do this. This is a very important technique. In the direct ELISA, you're detecting the antigen directly, therefore direct ELISA. So your sample containing antigens mixed with the antibody, enzyme-linked antibodies react with the antigen, and then you detect they're detected by adding a substrate for the linked enzyme. So, and then there's a color product produced, and that's how you can tell whether something's positive or not positive. In the indirect ELISA, we're detecting the antibodies. Basically, in HIV testing, for example, you are detecting the antibodies that somebody has made against HIV, against the virus, and that would be an indirect ELISA. In the direct ELISA, you would detect the virus itself, or the antigen, whatever is producing the reaction. Western blotting is also important. In Western blotting, you are separating a protein sample that contains various proteins in an electrophoresis gel. You're separating the proteins by size, and then you come in with an antibody that's directed or aimed against a specific protein. If that antibody finds its target, then you come in with a secondary antibody that's usually linked to an enzyme, and then you're producing a color reaction if, in fact, that first and secondary antibody have found their targets. Here's an example for a direct ELISA test. The example here is the pregnancy test, and so basically you're using monoclonal antibodies against human chorionic gonadotropin HCG. That's a hormone that's only present in pregnancy, and if the antibody finds that target, finds the HCG, so the monoclonal antibody is bound to the substrate here, then you form the sandwich, and then you can form a color reaction, and you will see. And if somebody's pregnant, then you will find sort of this line here in the test window. If the test is negative, then there is no line here in the test window on the bottom, and the upper line here is always a control window, so to make sure that the test is actually working. Here's a diagram that shows you the difference between the direct and the indirect ELISA method. So here on A, we have a direct ELISA. That means the antibody is absorbed to the well, and then we are adding the patient sample, and if it contains the antigen, then the antigen will bind to the antibody that's coded here in the well, and then we have a secondary antibody that is then binding to this complex of antibody antigen right here, and then the linked enzyme, this yellow part I guess, is producing a color reaction. So that would be a direct ELISA test. In the indirect ELISA test, indirect right here, the antigen is absorbed into the well. So like for example the HIV testing, we're absorbing the antigen into the well, and then the patient serum is added, and if the patient serum has the antibody, so let's say somebody was exposed to HIV, they formed the antibody, now they have antibody in the serum, and the antibody will now find the antigen that was coded in the well. And then the secondary antibody will find the primary antibody. That secondary antibody is linked to an enzyme, and it produces a color reaction. So the enzyme substrate will create a visible color change. Now, ELISA tests have proven very useful, and they're widely used for diagnosing certain conditions. And here's a slide about the future of diagnostic and therapeutic immunology, and there's a bright future ahead. Immunology and our knowledge of the immune system has proven very useful in many kind of diagnostic and therapeutic approaches. So the tests, they require less human judgment, and personnel, especially PCR and DNA probes, have been proven very useful as a diagnostic and therapeutic tool. They're relatively inexpensive and a simple test for developing countries, so they can be used quickly and inexpensively. And here, this is actually a really big point: disease prevention and therapy. So, of course, you can use your diagnostic tests for disease prevention. You can also use them to diagnose disease, and then use something that leads up to therapy. In addition, immunology has been used widely these days to develop monoclonal antibodies that can be used in cancer chemotherapy. It's very targeted approaches. Also, you have a bunch of new gene therapy approaches, and that will be involving CAR T-cell therapy, where the CAR stands for chimeric antigen receptor T-cell therapy. Basically what you're doing is you're taking T-cells from a patient and you are genetically modifying them. So it's basically a gene therapy that you're doing on the T-cells to teach the T-cells what receptors they should be going for in order to destroy certain cells. So it's used for cancer chemotherapy or cancer therapy in general, where you're taking a patient's T-cells and you're teaching these T-cells and reprogramming them to go after the receptors that are found on that patient's cancer cells. Once the T-cells have learned to recognize the surface structures that they're going for this cancer that the patient has, then you're reintroducing this new T-cell population that has learned something new and will go after those cancerous cells and destroy them in the patient, and has proven very successful. This is sort of the future of cancer therapy, to individualize therapy and using immunotherapy.